Hello, it's my great pleasure to talk to you about the future of IVF, in particular from a laboratory perspective. And these are my disclosures. I think it's important when we consider what we're going to accomplish in the future, the dynamics and complexity of life, that is the system in which we're working and trying to translate into the laboratory. This schematic shows the first week of human life, and it's highly dynamic, highly complex, the embryo changing its morphology, its physiology, its metabolism, and indeed its gene expression. And all the while, it's consistently interacting with the female reproductive tract. And furthermore, it's moving within the tract. So it's in close proximity to the mother, and at the same time, receiving signals and being moved by the female tract. And indeed, that's a cross-section of the human oviduct below. And you can see the close proximity in which the developing embryo will be to the maternal environment. And then we consider what we do in vitro, and you can see it's very dissimilar. Even though we've got more physiological with our culture media, we're still growing our embryos on polystyrene plates. And with the advent of tissue culture moving into time-lapse, we are now growing our embryos in microwells, but they're still polystyrene wells in a static environment. This gorgeous picture is taken from the early 60s with Bob Edwards and Jean Purdy. And in the background there, you can see their incubator, and inside that incubator is a glass desiccator. And that glass desiccator was used to create a stable environment in which actually Louise Brown was cultured and grown. Within 40 years, we've gone from those kinds of environments to this. So it leaves you asking the question, what can we expect in the future? And what I'll talk about is advances in microfluidics for how we prepare sperm and how we can diagnose sperm too. Advances in a thing called microfabrication or 3D printing that has led us to create devices specifically for oocytes and embryos to facilitate better ICSI culture and vitrification. We have to consider towards microrobotics and automation of the lab, and indeed looking to how do we better select embryos. And I predict again that we'll use biomarkers of health through sampling the media, or perhaps using different microscopies to analyze the health of the embryo. And of course, artificial intelligence will see its full potential within the IVF laboratory, not only from gamete and embryo selection, but how we monitor the laboratory, and indeed how we better use all the information that we're going to collect on each individual embryo as it develops and differentiates. So when we consider microfluidics, this is basically the behavior and precise manipulation of fluids in sub-microliter levels, very small volumes. And when you get down to these small volumes, fluid doesn't behave in a typical turbulent fashion, but more it exhibits a laminar flow. And these laminar flows have been used for many, many years, and we didn't even realize it, for example, within an inkjet cartridge to create precise delivery of fluids. In biology, microfluidics is used all the time in the phloem and xylem of plants, all the way through to our vascular capillary beds. And remember, at that capillary bed level, you're moving single cells, single erythrocytes through there. And again, you're seeing very, very small volumes of fluid being moved. And fluid behaves very different at these low volumes. And we can use that to our advantage. If you want to use microfluidics and laminar flow, it's an excellent process for separating immotile from motile cells. In this case, motile sperm cells from immotile spermatozoa or white blood cells. And indeed, 20 years ago, this was proof of principle was first shown by Shu Takayama and Gary Smith, whereby they used laminar flow system to show that only motile sperm could swim out of one stream into another. So the proof of principle was very exciting. But it's taken many years for these rudimentary systems to enter our field. And now there are many types of microfluidic devices being developed and indeed available commercially for selecting sperm. And what's really exciting, particularly in this paper by Jean-Pierre Palermo's team, was that you could use a microfluidic-based system to select sperm. And indeed, those sperm have a higher genomic integrity. And we and others have gone on to show that by using these such devices, and even more complicated microfluidic devices, you can select sperm with very low DFI. And that we hope will translate to much lower pregnancy losses. But beyond selecting sperm, the field of microfluidics and diagnostics is exploding. And in this review, which we published in Nature Reviews Urology just a few months ago, we exemplify how microfluidics can be used to do on-chip diagnosis, in this case, of extracellular vesicles. So we can look at extracellular vesicles in the epididymis, the epididymosome, or within the prostate fluid. So you can look inside all of these fluids and look for diagnostics, which is incredibly powerful. Where we see this all going is a multi-microfluidic-based reader, whereby raw semen is simply placed onto a chip, and that chip will sort out the sperm, as well as give you the ability to do CASA. We'll be able to filter the sperm, select motile sperm, sperm with low DFI, and do all kinds of cell surface markers. And at the end of the day, you get a readout of the health of the semen sample as well as the sperm. So these are the kinds of things that will be coming in the next few years. And indeed, when it comes to microfluidics, we did a proof of principle to show that you can actually use microfluidics to manipulate nano and picoliter amounts of fluid, in this case spent culture medium, and we're able to use an on-chip analysis to analyze the levels of metabolites in culture medium. And in this particular example, in collaboration with MIT in Boston, we were able to quantitate the uptake of glucose pyruvate and the production of lactate by single mouse blastocysts. But where things get very exciting is the consideration of a new technology called microfabrication. And why I want to talk about this is that when we look to the future, this was a review that I wrote 30 years ago when I was considering where I thought IVF was going then. So it's obviously something I spent quite a bit of my time over my career thinking about the future. And in this review in '94, I predict the rise and the use of blastocyst culture and the move to single embryo transfer, and in fact, moving to trophectoderm biopsy. I also predicted that we would be moving towards perfusion culture. Instead of using one media or indeed sequential media, we could put embryos in an environment where we could expose them to gradients of nutrients over time. So rather than just stochastic media, we could have a continuous flow of different environments, which would mimic more closely the oviduct and uterus. And of course, we got very excited about that. But sadly, the technology, the engineering wasn't sufficient to support that concept. So here we are 30 years later, and we still don't do it. However, that's about to change with the advent of 3D printing. And before I talk about the benefits of that, I just want to talk about this one paper, again, Shutakiyama, where they developed a microfluidic system to move embryos, in this case, agitate embryos using a Braille reader. So basically, there were pins that pop up and down like a Braille reader, and they agitate the fluid within a microfluidic chip. And Gary Smith went on to show that using that system, in these data, we can see over here, we have a microdrop containing mouse embryos, and this is the blastocyst cell number. And this is putting embryos in a microfluidic chip here, but it's just static. So the embryos are just sitting in this PDMS chip. But then when the Braille chip is activated and the embryos start to agitate, you can see there's not a replacement of media. What this represents is the embryo has been agitated and there's a doubling in cell number, almost a doubling in cell number, very close to the in vivo. So what this shows us is by simply moving the embryos around, which destroys any unstirred layers around the cells in culture, could facilitate better embryo development. So now I want to introduce you to 3D printing at a level that we could have never imagined possible, because this is a process using 2-photon polymerization lithography. Basically, lasers are used to solidify solutions in situ down at the submicron level, so you can create incredibly high resolution in non-embryo, non-gamete toxic polymers. So you can see here, this is the Eiffel Tower, and this is just 100 microns across. So you can imagine this would be a bit bigger, a human egg, just a little larger than that. And over here, you can see the bull, the teapot, and the gecko, and this is 1 micron resolution. So they're about the size of a sperm. So it's amazing the resolution that you can get down to. And now we can create devices. And this is the first report that we published in Scientific Reports earlier this year, whereby we created a device for microperfusion of cells. And this was work done with my longtime colleague and friend, Jeremy Thompson, whereby we used 3D microlithography to create chambers in which we could put our embryos, and then we could, in this case, we called them the cradle, and these cradles can then be docked into a garage, which is perfusion ready. So to better exemplify that, if I show you some of the schematics from the study, here we have basically was our cradle, and it's modified somewhat since we first started, and the egg or the embryo sits in the cradle, which then gets docked now into this nest, and you can perfuse across the embryo. And interestingly, when the flow is on, the embryo moves, and when the flow is turned off, it moves back. So simply by turning on this pump and off, you can get movement of the embryo. The other thing is, the flow rate of this is in nanoliters. So there's no shear stress across the embryo itself. So this is what it looks like in principle. We have the cradle that docks with the nest. And then you can attach culture media and monitor gradients and nutrients as the embryo is placed in the cradle, it docks in the nest, and then you can perfuse with nanoliter proficiency across the embryo. Absolutely remarkable. And if I show you now some video, here we are. The pump is on here, so the embryos are here. But when the pump is turned off, you'll see that the embryos move. So here we go. The pump is on, and at a certain time now, the pump will be turned off, and the embryos will move back. There we go, pump is off, and the embryos move, and in about 3 seconds, we'll turn the pump back on, and you'll see the embryos move across. So we're able to agitate the embryo, keep it moving, there we go, and then we'll see across the flow, the red, which is representing the medium, it's actually a food colorant, so you can see, but the red is going across and nanoliters, and it shows that the embryos are now being perfused at that high resolution. Okay, so now we move into automation. There's been several talks and discussions, and indeed pilot studies on micro-robotics, and these are two excellent examples of micro-robotics being used to immobilize sperm for ICSI. And more recently, there's a paper in RBM Online with Nueva Costa-Borges and John Zhang, and of course Santiago Monet, where they've looked at the first babies born using automated ICSI. In this case, they use a computer-aided design of ICSI, and this is the piezo drill. So this is the graphic of what it should look like, and here's the actual device. It wasn't fully automated, but it did show that the ICSI robot with high proficiency could be used in animals and in humans when operated by inexperienced personnel. So this is absolutely getting to that level. The next step is moving towards automation. And we believe that if you incorporate such a thing as automated microinjection with devices such as can be created using 2-photon polymerization, you can actually create devices that can move the eggs or the embryos within an automated system. And in this case, a device was created to house the eggs for ICSI. So rather than being held on a holding pipette, the eggs themselves are held into a 3D printed device, which means you do not need the holding pipette or that second micro manipulator. Rather, the whole dish is moved using the stage. And the eggs themselves can then be ICSI'd in situ. So again, these technologies are really paving the way towards automation, and all of which will streamline the workflow. And in this case, we believe will reduce stress on the oocyte itself. So a lot of that was recently published in Fertility Sterility on making and selecting the best embryos in the laboratory. But now I want to move over to non-invasive biomarkers of viability and health, how we're better going to select embryos. So for the past 40 years, we've created very elegant grading systems for every stage of development in the human embryo. But what if we want to look beyond morphology and morphokinetics? In this case, what if we were able to sample the medium and measure in real time the uptake of glucose pyruvate, amino acids, and the production of metabolites such as lactate? And indeed, in the environment surrounding the embryo and the medium, look for DNA, microRNAs, exosomes. Would that ultimately lead to non-invasive PGT? So this is something that, from a metabolite point of view, I've been very interested in for 40 years now. Why would you be interested in the metabolism of embryos? Well, the metabolism of any cell is fundamental to its survival. But during the pre-implantation period, the embryo undergoes dramatic changes in its nutrient utilization, going from a carboxylic acid-based metabolism to one based on glucose post-compaction. And what we know is if that transition doesn't go ahead, or something happens and the embryo loses control of its metabolism, then viability is lost. So hence, metabolism is actually a very useful indicator of the health of the embryo. These data showed that the glucose uptake of an individual human embryo from day 4 to day 5 was related to pregnancy outcome. In this case, those blastocysts that went on to give a pregnancy following single embryo culture, and we simply analyzed the medium surrounding the embryo to measure the glucose, had a higher glucose uptake. And those that failed to give pregnancy had a clearly significant less utilization of glucose. We've then gone on to repeat this in a bigger data set. But as well, we're able to look at how metabolism was also linked not only to the Gardner grade, but to morphokinetics and artificial intelligence. So the first thing, again, we repeated was that glucose uptakes were higher in those blastocysts that go on to give pregnancy. So that's consistent with the previous study I just showed you from 10 years earlier. When we looked at glucose uptake, we found that it was related to the alphanumeric score, the Gardner grade of the blastocyst, the KID score, and also the artificial intelligence score. So what we're seeing is that metabolism is indeed related to a lot of these parameters that we've been using to grade our embryos. Interestingly, when we looked at amino acids as well, again, looking at 2 different cutoffs of artificial intelligence, but a similar thing happened with the Gardner grade and indeed morphokinetics, was there were fundamental differences in the way embryos were utilizing amino acids, which implies it's not necessarily that poorer quality embryos are using just less metabolites. It's actually, in the case of amino acids, they could indeed have different physiologies altogether. Now that requires sampling the medium and using quite sophisticated methods of metabolite analysis. What if we could replace that with light? And this is a review that Denny and Sakis and I wrote a few years ago, looking at novel microscopies, in particular fluorescence lifetime imaging and hyperspectral. And the premise of this is that you can use these microscopies to quantitate intracellular levels of cofactors which are linked to metabolic state. In particular, on the left plate in blue, we were looking at nicotinamide adenine dinucleotide, or NAD. That's typically cytosolic and is related very heavily to glycolytic metabolism. In B, you can see that we were looking at flavine adenine dinucleotide, or FAD, and that reflects mitochondrial metabolism. And indeed, you can see the green is particularly well sequestered into little drops, which are the mitochondria in a perinuclear pattern. So this is really interesting. Can we use light? A few years ago, I was asked to write an editorial on the Gardner grade for ASRM's 75th birthday gift in FNS. And in it, I wrote an important point, and that is aneuploidy in somatic cells results in a phenomenon known as aneuploid stress. Aneuploid cells have an altered karyotype. We all look for that, and then we've been looking for that in PGTA for many years. But the reality is that altered karyotype leads to an altered pattern of gene expression. That altered pattern of gene expression changes the metabolism and proteome of the embryo. So an aneuploid cell exhibits physiological or aneuploid stress. In other words, they could have compromised metabolic states. And this is a beautiful paper using hyperspectral microscopy, again, using different wavelengths of light to quantitate the metabolic signal from an embryo. And in this case, Kylie Dunning's group from Adelaide not only looked at mouse embryos, which they induced to be aneuploid using chemicals, but also looked at human fibroblast cell lines and those that they knew were euploid or aneuploid. And they showed by light they could differentiate metabolically aneuploid and euploid cell lines. And then this tremendously exciting paper came out from Denny Sakis' group in Boston. This was using fluorescence lifetime imaging, as I described in the earlier slide, but looking at euploid and diploid embryos. So the aneuploid embryos are in the dark blue and the euploid are gray. And what they showed is when they looked at the entire blastocyst, there were differences in metabolic states of these embryos using fluorescence lifetime imaging. They were then able to look within the embryo at the inner cell mass and showed there were great significant differences between the inner cell mass of aneuploid and euploid, and again, between the trophectoderms. So this is really fascinating. It's using light to differentiate aneuploid and euploid. Now, whether we can use this to rank embryos is a tremendously exciting proposition. I also want to introduce with this a whole paradigm shift in our understanding of development, and that is a thing called metaboloepigenetics. Metaboloepigenetics is the relationship between metabolism and the epigenome. And this comes about because a lot of the cofactors that are used during metabolism are actually regulators of the epigenetic state of a cell. And one in particular, NAD, that I've referred to, can be picked up by phlegm, is a very powerful regulator of the sirtuins and their acetylation state. So can we use light to actually ascertain the metabolic and indeed the epigenetic state of an embryo? Is something that we await to see if we can do, but it's a tremendously exciting prospect. And of course, we couldn't talk about the future without talking about artificial intelligence. This was the first paper to develop an artificial intelligence through deep learning to show that we could actually use AI to select human embryos. And this has now undergone a prospective randomized trial, and those data have been submitted for publication. And indeed, it's been shown that fully automated, in this case, the IDA score, was shown to perform at least as good as state-of-the-art manual selection models. So this is really interesting. It's saying that artificial intelligence could be just as good as a human embryologist, not better, but not worse than a human embryologist. The other thing it does, it removes the intra-operator variability, gets rid of that subjectivity that we're all familiar with when it comes to grading our beautiful embryos. A lot of people are worried, though, that artificial intelligence is going to replace human intelligence. But when you actually talk to artificial intelligence engineers, they always say, actually, the strength of AI is to work with human intelligence, where there's synergies in terms of outputs. And indeed, these two very elegant papers show that the combination of AI and conventional grading provided the optimum output. And indeed, this paper has got to be one of the best titles of any paper I've read recently, Should There Be an AI in Team, by Charlie Borman's group, and again showing that the incorporation of AI for blastocyst selection can enhance the performance of a trained embryologist, but working with the trained embryologist. So AI is not going to replace embryologists. Embryologists using AI will replace embryologists. And of course, we can use AI now to look at sperm selection. And this is really a seminal paper whereby Javadi and co. showed that they could use AI to examine and select individual human sperm based on WHO criteria. This could be a game changer in terms of diagnostics. Some of our own recent work we presented at the ESHRA this year showed that we could create AI to look at sperm in testis biopsies for ICSI. So we could train AI on raw images from a testis biopsy and move all the way through to side-by-side testing against an embryologist. And this is what we're able to show, is the time per field of view over here. This is the embryologist, and this is how long it took artificial intelligence to find the sperm. When you compound all of the fields of view within a testis biopsy, it took the embryologist 5 hours to complete the search for sperm. When you compared that to how long it took the artificial intelligence, the AI only took 10 seconds. So the good news on that is that it could save a considerable amount of time and upper body stress in the embryologist searching for those elusive sperm in a biopsy. But was it as accurate? In fact, AI missed 184 of the sperm in the sample, whereas the embryologist missed 320. So actually, the AI was more accurate as well. So time-saving, increased accuracy, this would be a phenomenal addition to our armory when it comes to looking through testis biopsies. And of course, AI doesn't stop there. AI can be used to sit in the background and monitor all of our KPIs. And I think this is one of the great powers of AI when you've got so many variables, hundreds of variables in each IVF lab, all been monitored by artificial intelligence. So our KPIs have been updated in real time. And that way we can avoid any problems before they actually manifest. So we propose we could take all of this and combine it into a future laboratory in where we don't grow embryos in dishes anymore. We grow them in microfluidic devices whereby we can perfuse the embryos. We can then sample the culture media, look for biomarkers, look for cell-free DNA. Rather than use conventional microscopy, perhaps we can use time-lapse as well as hyperspectral and/or FLIN, or perhaps optical coherence tomography may come and give us even greater resolution. So for each embryo, we will not only have image analysis from different microscopies, we'll have biomarker analysis, and artificial intelligence will then help us create better output scores for each of our embryos. So in conclusion, I think microfluidics is an excellent platform for sperm diagnosis and preparation. We are seeing it being used for preparation. In future, I think this is going to be used for diagnosis. These fields in microfluidics are rapidly developing, which is truly exciting. The novelty of microfabrication provides for the first time the opportunity to investigate perfusion culture, to create dynamic environments in real time. And now this is at the research stage, but over the coming years, we'll be able to determine whether perfusion actually gives us an advantage over embryos just sitting in a drop for several days. Microrobotics could really help move towards standardization of technically demanding tasks such as ICSI. And again, using microfabricated devices will facilitate that translation as we create carriers that we can have in the dishes to facilitate the movement around. New microscopies such as FLIM and hyperspectral could provide new insights into the metabolic state of embryos. And I think that's really, really exciting. And we'd be really keen to see whether those metabolic states reflect the carrier type. We know that you can analyze the media, and that is very exciting when it comes down to looking at viability. But could we use this in the future to assess the epigenetic state of an embryo? It's potentially possible. And again, the new microscopies could help us do that. Artificial intelligence is not just about embryo selection. It offers so many more advantages to us in the laboratory. We should not be afraid of it. We should embrace it. It's not going to replace us. We're going to work with it in synergy to enhance what we do, and in fact, be able to use far more data than we currently do to diagnose our embryos. And evidently, through automation, through artificial intelligence, less time will be required, thereby making the labs more efficient. But we, as embryologists, will always be required. Embryologists will be able to do more cycles per annum as a result of this. But the reality is there's an increasing need for assisted conception. And with these advances, we'll be able to reduce costs and increase availability. So I think that's tremendously exciting. And a lot of what I've put forward in the ideas here, I published in this Countercurrent last year in RBM Online. The future is incredibly exciting. And I've pondered upon this for many, many years. What's really exciting about today is the technology that actually caught up with the ideas we've had for many, many years. And I think we're on the verge of seeing many of these new technologies being trialed and implemented in our IVF clinics over the next few years. It's a tremendous honor to have the opportunity to talk to you. Thank you.